Hello, everyone, and welcome to the Cost Action Sustained webinar series. My name is Saeed Ishaq Deliri, responsible for coordinating this session. Today's webinar is organized by Working Group 2, Plant Responses to Salinity, and we have the pleasure to hearing from our two outstanding researchers, Mr. Mohamed Abdelfadil and Mr. Pascal Mende. We will begin shortly. Please keep your microphones muted and feel free to use the chat for questions. We will have a short discussion after each talk. Now, I would like to kindly invite Professor Jutta Peppenbrock, leader of Working Group 2, to briefly introduce today's speakers and open the scientific session. Professor Peppenbrock, please go ahead.
Yes, thank you very much for organizing this webinar, Said. Yeah, and welcome to all participants. It's really great to see many people here. And it's also great that you somehow participate in this really great cost action that is actually led by Kate Neggarts, who is also here. Yes, it's the first webinar that we actually organize within this cost action. So it's new, it's the first time. Yes, actually, we invited two young male researchers to give an overview about their PhD thesis, or actually also part of a master thesis. And one reason is that actually our cost action is very much, let's say, dominated by female researchers. And therefore, we thought, oh, it would be maybe great to give also young male researchers the chance to present and to participate in this cost action actively. Yes, so we will start with Mohamed. He actually did his plant biotechnology studies in Egypt at Cairo University. Then he moved to Germany, to Berlin, and did his PhD at Humboldt University. But I think his main experimental work was done as the Institute for Horticulture in Großbeeren. And at this institution, he is actually also doing now his postdoc. Well, I'm saying postdoc. His PhD has not finally finished, so maybe it's also good practice. He will finish, I think, this summer. Yeah, and now we are looking forward for your presentation. He's interested in plant microbe interaction, and I think that will be also in the focus of the presentation. So the floor is yours. Just one point to the question. So we have only 5 minutes for a question, actually. So the presentation will last about 25 minutes. And you can write your questions in the chat or in the question and answer session. So thank you. Now the floor is yours, and it would be great if you can share the screen.
Yes. So I think you are able to see it right now,
Perfect.
right?
Perfect.
Yes. Thank you so much, Lisa, for your introduction. I'm happy to introduce my research work in front of you today with the title Enhancing the Tomato Salt Tolerance Through the Synthetic Microbial Community and Microbiome Transplantation from Halophytes. So in the beginning, and I want to say why. First, I want to mention why microbes. Microbes are the most experienced living organisms facing and adapting to fluctuation environmental events, and they have persisted for a billion of years under the harsh conditions. And they are not only habitat our guts, but also our plant parts such as rhizosphere, and the community of these microorganisms in the exist in a particular environment is the microbiome. And as we know that our agroecosystem face challenges such as salinity stress that impact on the agricultural productivity. And this stress is caused by different factors such as climate change, which also impact to the plant growth by inducing such as osmotic stress, ionic imbalance, and altering the soil structure. And we can imagine that nearly 10% of our world's land surface are already salt affected. But we have naturally salt-tolerant plant in our planet, the halophytes. These halophytes plants have evolved with various adaptive salt mechanisms, salt-tolerant mechanisms, to adapt in high salinity environments. But these plants have not been adapted alone. They have been co-evolved with their microbiota. And this interaction between the plants and their associated microbiome results from millions of years of their co-evolution. So these plants also cooperate with their associated microbes with strategies such as cry for help, this to elevate the stress by they are change their root oxidation chemistry to assemble a healthy and a beneficial microbiome. Therefore, we hypothesized that this microbiota that resulting from millions of years of co-evolution with the halophytes may offer a potential to assist the non-halophytes plant in adapt to salinity stress. And the microbiome strategies, such as whole microbiome transplantation and the function and the synthetic community, that is the specific taxa with effective functional traits that can mimic the whole microbiome functions. But these microbiome-based strategies facing multiplied challenges, such as selecting the candidate microbial species with the complex communities. And also, we need to determine the suitable donors, like which halophytes, which plants. And also, the second challenge, or one of the most important challenge, the suitable delivery system, that we need suitable delivery system that allow to control the microbiome that you will choose, and also maintained under the changing condition during transfer to ecosystem, and also able to reproducible this microbiome inoculum that if you choose from the halophytes two times or repeat it multiply times, so it's still a challenge. So our main aim was to present or introduce a method for first to capturing and preserving the rhizosphere microbiome in situ, and second, select and identify the key functional microbes in the microbiome of halophytic plants, and third, validate the selected microbiome and test if they are able to improve the other plants to mitigate the stress, such as the crop plants. And for the suitable delivery system, as I mentioned, we need suitable delivery system able to capture the diverse type of microbes and also able to preserve this type of microbes for long term to transfer to the target ecosystems. And the idea emerged and came from the ancient Egypt. As when the scientists discovered the tombs, they found some living microbes still survival in the clay matrix of the ancient pottery for thousands of years. So we decided, why not use the same type of clay as a low-tech and low-cost methodology for capturing and storing the living root microbiota. So we formulate this type of clay, such type of this clay in beads, and then we tested the ability of this material to capture the rhizosphere microbiota by we added this clay material beside to the plants during their growing, and then we used the culture dependent and the culture independent to investigate how this similarity between the community that captured in the clay material and between the root, between the plant roots. So we interestingly found that these clay able to capturing the community composition with similar structure to the plant roots. And we decided also to test this type of clay also to preserve different type of microbes. We decided why not also test it to preserve like representative microbes such as gram-positive and gram-negative and fungi. And interestingly, we found that the clay beads, this clay method, able to preserve or successful to preserve these three types of microbes up to 1 year, even at ambient room temperature. So now we have this suitable system that can allow us to apply the microbiome with ability to reproduce inoculants through multiply preserved copies of the microbiome that preserved in this type of clays. However, we have now still two fundamental questions remains. What unique properties should be embedded in the microbiome to select? Like which taxa that can be responsible for the function to mitigate the stress, and also which donors of the plant that can be suitable to take this microbiome to transfer to another plants. And the second question, can this specific microbiome able rapidly to promote the adaptation of the recipient plant traits under the stress conditions? Therefore, we conducted a meta-analysis study we investigate the variation in the microbiome structure and to identify the taxa that highly associated with these plants. And we collected the published microbiome raw data from different halophytes plant and non-halophytes plant worldwide, such as the crop plants, such as maize, rice, wheat, and tomato. And we collected 15 microbiome studies for 40 plants. And we here found interestingly results that we found based on the bacterial genus and community structure that these plants that grow, we found two different groups of halophytes, they are separated. Interestingly, these groups separated based on the salinity in the rhizosphere, as we found that the group of the halophytes that grow in high salinity conditions, they have a different community from the halophytes that grow in low salinity conditions or from non-halophytes plants. Even they have the same plant species. So here that we identified that the salinity represent a factor for controlling the rhizosphere microbiome, also between the same plant species. So now we can, if we want to select a specific donor, we need to have the specific environmental condition for this plant. For example, they are growing in high salinity condition or low salinity conditions, but they have a different community. That means it's not all of the halophytes have the same community structure. So our next step to identify the marker taxa that associated with the group of the halophytes that grow in high salinity conditions. And we used 3 different approaches to identify the microbes that maybe have this function to mitigate the stress that associated with this group. That we used the core microbiome, the taxa that are shared across all the plants. So here you can see here we have the 3 groups of plants, and for the high halophytes plant, they have a distinct core taxa which are around 15 or 16 bacterial genera that not present in the low halophytes or non-halophytes. And also, we used the key hub taxa to also identify the taxa that responsible about the stability of the community structure in this high halophytes plant. And also we identified the taxa, the important taxa or differentially abundant taxa using the machine learning algorithm like random forest. And then we had like this, the shared or the key taxa that shared between the three approaches that shared between the whole plants in the high halophytes group and identify what is the key taxa in this group and the important taxa using the machine learning algorithm. And then we made like alignment. Now we have the genus level, which now it's challenge how we can use it, because under each genus we have a lot of species. So we made like alignment between this genus level with the available bacterial species that in the culture collection, like this MZ culture collection. And then based on these taxa or this species that we have now, we conducted a synthetic community. And I want also to mention something here. These taxa that are unique for this high halophytes group are, most of them are halophytic taxa. And then we have now, as I mentioned, the method able to capture the whole microbiome and able to preserve for applications such as whole microbiome transfer. And also we identified taxa that only unique for the group of the halophytes that grow in high salinity conditions and constructed from it a synthetic community. And then we went to two-way. So the first one, we chose the Salicornia group plants, which is one from the group of the high halophytes group plants that grown in high salinity conditions. And we used our method, the clay beads that I presented in the beginning, to capture and store the community for whole microbiome transfer. Also, for the second way, we went to based on the syncoms that we identified based on the meta-analysis. And now we want to validate the effect. So here you can see the workflow of our experiment. So as I mentioned here, we collected the rhizosphere microbiome from Salicornia, and here we collected the microbiome from two different sites, which named M1 for a site for Salicornia and M2, and then the synthetic community that we constructed based on the meta-analysis from comparing the halophytes with the high halophytes plants with the low halophytes and non-halophytes plants. And here we inoculated two type of plants. We inoculated two type of tomato plants, salt sensitive and salt tolerant. And the aim here to use the salt tolerant to test if this microbiome application able to increase their salinity tolerance threshold to mitigate the stress. So for example, if they are able to this salt tolerant to mitigate until 100 millimolar NaCl, if we add our microbiome application able to increase their salinity adaptation. And also here we tested these two type of tomato plants with 3 salinity conditions: 0 millimolar NaCl, 300 millimolar NaCl, and 500 millimolar NaCl. And the aim here to use these high condition of salinity to see the effect or the real effect of the microbiome to mitigate or to improve the plant adaptation to stress conditions. And we had here two hypotheses. The first one, the microbiome application, the whole microbiome transplantation or rhizosphere microbiome transplantation, and synthetic community able to enhance the growth of salt-sensitive and salt-tolerant plant. And the second one, if these application or microbiome applications able to enhance the colonization of the tomato plants by halophytic community in response to salinity stress. Interestingly, we found that these microbiome application able to enhance the growth of the tomato plant parameters, such as the shoot dry weight content. So here, interestingly, that these two plant species, they are responded differentially. But here you can see how this, here's the sensitive variety and tolerant variety, and here's the salinity applications, 0 millimolar, 300 millimolar, and 500 millimolar NaCl. And here our treatments. Here you can see the control with this red color, and you can see how this microbiome application significantly able to enhance such shoot dry weight parameter, and also able to significantly improve the chlorophyll content in the two variety, and also able to decrease such the sodium accumulation in the plant leaves compared to the control plants. So it's reduced the sodium accumulation in the plant leaves. So we want to also look at what happened to the tomato plants after we added this inoculum or this microbiome inoculums. Interestingly, we found that this microbiome application able to shift the native rhizosphere microbiome of the tomato plants. And you can see here how the control plant, they are clustered significantly away from the other treatments from the microbiome transplantation from site 1 or site 2 and sensitive community that we added to the plants under the 300 millimolar NaCl and 500 millimolar NaCl. And also that's happened in the tolerant variety, that this microbiome application successfully modulated the community composition of the tomato plants. But to which direction this shift or improvement in the microbiome composition? So here we wanted to test if the syncom that we applied or we added to the tomato plants able to colonize the rhizosphere. So here we added like 9 species from the syncom, and interestingly, under the increasing of the salinity, we found that this microbiome able to establish, or this bacterial taxa that we added, able to significantly establish to the tomato rhizosphere plants for the plants that inoculated with the synthetic community with uniquely taxa such as Legionella and Marinobacter, which are halophilic and marine taxa. And also that's happened in the tolerant species, which interestingly able the taxa, the 9 taxa that we added, more than of the species that are able to colonize these tomato plants. And also uniquely for such Legionella, Martella, and Rosivivix, which are also marine taxa. So here we can conclude that this syncom taxa can successfully able to colonize and dominate significantly in response to high salinity in the tomato rhizosphere plants. Also, for the root rhizosphere microbiome transplantation that we transferred from the Salicornia Europa. Interestingly, we found that the high contribution from the inoculums that we added increased with the increasing of the salinity under the 300 millimolar and the 500 millimolar in the sensitive and tolerant variety, with highest contribution of the genus from the inoculum, more than 50%. That means more than 50%, as you can see here, sourced from the inoculum are established in the tomato rhizosphere that happen in the microbiome for the plants that inoculated with the microbiome from Salicornia from site 1 and also from the site 2 for the sensitive variety and also for the tolerant variety. So now our current step, we performed like shotgun proteomic analysis of the tomato plants that's subjected to salt stress and treated with this different microbiome. And you can see here how this also the two plant variety, they have also regulating significantly with different proteins, with also interestingly some unique function or some unique proteins for the microbiome applications, such as syncom and microbiome from that transferred from site 1. And also this is for the plants that the host that inoculated with the microbiome for our experiment. And also we did also this proteomic analysis for the microbiota around the rhizosphere for our treatments. But the analysis is still ongoing for both this plant and for its associated microbiota. And here we are aiming for our next step to identify which proteins that are regulated or down-regulated with these microbiome applications compared to the non-inoculated control. So finally here, the take-home message that halophytes represent a repository of robust microbes that poses beneficial traits for other sensitive plants. Also, the in silico identification of shared pattern associated with the plants with a similar environmental condition can help to design effective syncom that can able rapidly enhance the plant resilience to stress conditions. And the third one, that microbiome-based applications such as whole microbiome transplantation and syncom can rapidly improve the adaptation and resilience of non-halophytic plants such as tomato to salinity stress. And thank you so much for your questions, for your attention. Sorry.
Thank you very much for this brilliant talk. It was really interesting, and I'm sure that you will deserve your PhD in the near future. Yes, there are already some questions in the chat. I'm not sure whether you can read them yourselves, but so there's one question. So on the experiment where you inoculated two plant species with different inoculation and treated them with different levels of salinity, the inoculated plants did better than the control plants. However, it looked that all the inoculation worked better then there was no difference between the different inoculation. Is that correct? So could you observe any difference between the different inoculations?
Yes, thank you for this good question. But here we expect the microbes that we added for these inoculums behave the same, like improve the plants. And there are different mechanisms for these microbes that in this community can help the plant to mitigate the stress. So this is our aim from the beginning, that we hypothesize that these applications or this inoculation have special microbes that can able to improve the plants. So interestingly, we found we didn't observe it like high variation between the inoculations. And now we want our next step also, our currently step that we already finished, that we identified that also some shared microbes between these three inoculations. These shared microbes can also be the reason for this similar very phenotypic similarity between the plants that inoculated with microbiome application.
Okay. Did you also check actually the fruits, or did you just, I was not quite sure. I mean, that would be also interesting to analyze fruits and whether there's any changes in the aroma of the fruits.
Actually not, because the plant's really stressed, because 500, it's equal like seawater.
Yeah.
So here we, as I mentioned, we use this high impact of salinity to test
Yeah.
if the microbiome will work or not with
Okay.
our
Yeah.
applications. So,
Mhm.
for example, if we want to test the tomato with this microbiome application for the future, we can
Mhm.
decrease this salinity to test like and take the fruits or the yield from these tomato plants.
Mhm. I mean, sometimes mild stress also increases specialized molecules and therefore also aroma. So that might be another beneficial aspect. Okay, there's one other question. It is asked, did you look into the pathogen control by the microbes from halophytes?
Yes, it's a good question. We did an experiment in the beginning using this microbiome transplantation because we expect when we transfer the whole community, we can also transfer some pathogens with us. So
Mhm.
here in the beginning, we tested if they have any impact from the pathogen, like preliminary experiment, on when we applied these applications. And interestingly, we found like not disease or indication for disease.
Mhm. Okay, thank you very much. I don't see any more questions, and actually, there's also not so much time. So, thank you again. Really, really interesting work and a very promising work. I think, yeah, sailor and agriculture will also contribute a lot or benefit a lot from your work. So, thank you again.
So, thank you so much. So,
So I think now we can, yeah, switch to the second speaker. That will be Pascal Mende. Actually, Pascal finished his bachelor and master thesis at Hannover University in Germany and started his PhD just recently. And therefore also to involve young researchers, we decided that it would be great to have a talk from him. Pascal, we cannot see it in the presentation mode yet, so maybe you can modify again. And you will see that, yes, he is interested in quite an extreme halophytic plants, and he's not only interested in sodium chloride as a salt, but also in other salt that might be present. Now it's perfect, and we see your presentation in presentation mode. So the floor is yours, Pascal.
Yeah, thank you very much. First of all, thanks for the opportunity to share my results of my current research in this webinar today. In the next 20 minutes, I will talk about the plant you can see here on the left side. This is Strombocarpa strombulifera. It's a halophytic shrub which grows in semi-arid and arid regions of North and South America, and it's very common in the central region of Argentina. And as you can already see, these soils contain high amounts of salts, which makes these plants a good model to investigate salt tolerance mechanisms in plants. Yeah, before I dive deeper into the topic, let's first introduce our working group. I'm a PhD student at the Institute of Botany at the Leibniz University of Hannover. And here in this picture, you can see our current working group. You can see Mrs. Papenbrock, and in the middle, you can see me. And as you can see, we are a very young team with a lot of bachelor and master students. And one of our research topics is our halophytes. And on my left, you can see Tim Sprengel. He's a bachelor student who is helping me at the moment with my current experiment. And to my right, you can see Abdelfadil. He was the supervisor of my master thesis. And he's working with Salicornia, especially Salicornia europea. And he's trying to establish this plant as a new crop plant. Yeah, and now let's dive into the topic. As you all know, soil salinity or soil salinization is a serious problem for our agricultural production. And at the moment, there are more than 800 million hectares of soils worldwide affected by salt, and this is increasing year by year. And what are the reasons for this soil salinization? There are on the one hand natural sources like the climate. So in arid and semi-arid regions where there's a high evaporation of water, this leads to accumulation of salts at the soil surface. Another natural source is the chemical weathering of parental rock and also seawater contact. On the other hand, there are secondary drivers or human-made drivers for soil salinization, like irrigation or the use of fertilizers, and the climate change worsen all these effects. So now is the question how we can counteract this. And there's a pretty nice review article where the authors discuss two strategies to create the plants of our future. The first strategy is that you domesticate already tolerant plant and try to establish a crop. This is related to the topic Abdelfadil from my working group is working with. And second strategy is that you find salt tolerance genes in halophytes and introduce these genes into our current crops to improve the resistance. And another method is to use microorganisms to improve the plant stress resilience, like Mohamed was talking in his presentation. Yeah, and for the second strategy, Strombocarpa, strombulifera serve as a promising candidate. Here you can see this plant. It's a spiny shrub with a heat between 0.5 to 1.5 meter. And as I already said, it grows in the central region of Argentina, where the proportions of sodium chloride and sodium sulfate are nearly the same. And previous studies have shown that there's a high tolerance against sodium chloride, up to 1 molar. And on the other hand, there's a strong growth inhibition if you treat these plants with sodium sulfate. Yeah, and in previous studies, you can see in the physiological and biochemical analysis these two different effects. So a tolerant phenotype if you treat it with sodium chloride, and a toxic effect if you treat the plants with sodium sulfate. But what is missing at the moment is that you investigate this question on a molecular level. And this was the start of my master thesis, where I tried to establish a repeatable protocol for gene expression analysis in this plant to, on the one side, find genes, or to later find genes for salt tolerance, and on the other hand, to explain these toxic and tolerant phenotypes. Yeah, and for qPCR analysis, you first have to extract RNA from your plant. So I had to find a working protocol to extract RNA of good quality. Then I synthesize cDNA, and in the end, you can use this cDNA library to perform a qPCR analysis, and therefore you have to design primers for reference genes and also for genes of interest. Here you can see my experimental setup. First, I germinated my seeds, and then I transferred the seedlings into these hydroponic boxes, which containing 25% Hopeland solution, and let them grow for 40 days. And all 2 weeks, I changed the Hopeland medium. And yeah, here you can see the plants during this time. So after 1 week, 24 days, and 5 days before the treatment and the harvest started. On day 40, I started with a treatment. Here I used 3 different treatments: a control treatment with just water and 2 salt treatments with sodium chloride and sodium sulfate. And then I harvested the plants at 8 different time points. So the first harvest was directly before the treatment started. And then I harvested plants 30 minutes after treatment, 1 hour after treatment, until 48 hours after treatment. And for each time point, I had 5 biological replicates. Yeah, so the next step was to extract RNA from these plant samples, which was not so easy because the leaves of Strombocarpa strombulifera contains mucilages, which you can see here in these pictures, this middle phase, which make it very difficult to extract RNA with a good quality. So I tested several different protocols, but in the end, I found a working one, which is based on CTAP PVP, and this helps to remove polysaccharides, which are the main structure of mucilages. Yeah, and so I was able to extract RNA of good quality and quantity, and also you can see here there was a good integrity, so I could synthesize cDNA from this RNA to prepare a cDNA library, and with this cDNA library I then performed qPCR measurements. This was also a challenge because at the moment there's no sequence data available for Strombocarpa strombulifera, only for related species like Prosobiscinaria and Neltuma alba. So I performed a primer blast with sequences of these both species, and the idea was to find primers which can bind in both species and to hit conserved regions so that these primers can also bind to Strombocarpa strombulifera. And yeah, it worked out in the end, and I was able to design primers for reference genes. Here you can see my 4 reference genes: actin, ubiquitin, elongation factor 1 alpha, and 18S. And also I designed primers for some genes of interest, which are related to salt stress response. So I've some primers for genes for transporters, so like NHX, sulfur, and potassium exchanger. I also designed a primer for ascorbate peroxidase, which is an antioxidant enzyme, transcription factor BZIP, and a heat shock protein, which serve as a chaperone under salt stress. Yeah, and here you can see my result on this slide, the relative gene expression for NHX. NHX plays an important role in response to salt stress in plants because it's a transporter located in the tonoplast, so the membrane in the vacuoles, and it helps to remove sodium from the cytoplasm and store it into the vacuole to decrease ionic effects, toxic effects, and also to maintain the osmotic balance. And on the slide, you can see on the left side the results for my leaf samples, and on the right side, the results of my root samples. And on the y-axis, there's always relative gene expression, and on the x-axis, you can see my 3 treatments, so control, sodium sulfate, and sodium chloride, and also my 8 different harvest time points from 0 to 48 hours after treatment. And as you can see in the leaf samples, there's no significant difference between control and sodium chloride treatment, but there is a significant increase of NHX expression in the sodium sulfate treated plants. After 8 hours, there's a significant increase. And yeah, here you can see again this the same pattern like in the previous studies. So there's a tolerant response or like a control response for the sodium chloride treatment, and there's a salt stress response for the sodium sulfate treatment. I also tested other genes of interest, for example, the ascorbate peroxidase This is an important enzyme for the antioxidant system, and it converts hydrogen peroxide, which increase under salt stress, into water. And also here you can see a significant increase for the sodium sulfate treatment. Same for the sulfate transporter 2.1. Here's a downregulation, a significant downregulation of these transporters under sodium sulfate treatment. And for these other two genes of interest, I couldn't observe this pattern like in previous studies, so there's not this toxic effect of sodium sulfate. What you can see if you analyze the proline content, proline serves as a indicator for stress in plant cells, and you can see there is a significant increase over the time for both treatment in both organs, and you can see there's a significant increase after 12 hours. So in my actual experiment, I also included last harvest time point after 72 hours to also investigate the late response to salt stress. But what is the result of my master thesis? I was able to establish a working protocol for qPCR analysis, and now it's time to analyze more genes of interest. For example, Tim Spengel, the bachelor student, want to analyze sulfur transporters to see if there's a specific response to salinity stress under for this sulfate transport, especially for the sodium sulfate treatment. And also I want to test more salts with more ions and ion ratios. Strombocarpa keeps a challenging research model due to these mucilages, but maybe this is also an interesting part of this plant. So maybe this is a strategy to overcome the high salinity in the soil where this plant grows. Now to my current project. I started beginning of this year with my PhD, and in my current experiment, I want to analyze 4 different salts, so sodium chloride, potassium chloride, sodium sulfate, and potassium sulfate, to see the different effects of these ions, and also to see if this toxic effect is due to the sulfate or it's due to the sodium. Then I'm planning to do some RNA sequencing analysis to compare sodium chloride treatments with sodium sulfate treatment to better understand these 2 phenotypes, and also to find new genes for salt tolerance. And I also compare this with glycophyte, with Medicago truncatula. Why I choose Medicago truncatula? Because it's the model organism for legumes, so there's a lot of sequence data, and the idea is that with this data it's easier to design primers for Strombocarpa, and also it's interesting to see the difference of these responses to these 4 different salts between a glycophyte and a halophyte. And if I can find some interesting genes during RNA sequencing, maybe also the next step would be to transform this into Medicago and see if this makes these plants more tolerant against salinity. And also I want to investigate the role of the mucilages and want to see if this is maybe a function, protective function against salinity stress. And at the end, 2 pictures of my current experiment. This week there was a harvest of my experiment. On the left side you can see the Medicagos, and on the right side you can see Strombocarpa. And I'm really looking forward to the results and to see the different responses of a glycophyte and a halophyte, and then later to see the results of the RNA sequencing experiment, and maybe to find new genes for salt tolerance, to transfer them later into Medicargo. That was my presentation. Thank you very much for your attention.
Yeah, thank you very much, Pascal. Perfectly in time, and really nice approach. So you got already 2 questions in the chat. They are kind of similar. So they ask, could it be that the constitutive expression of selected genes is already very high compared to glycophytes? Sorry, it's difficult to read here for me. No, not fact. And then there was it's a related question whether if you analyze gene expression already so early after you applied the salt, whether it's more like a salt shock, and whether you would expect that after some time maybe plants would have adjusted to the salinity. So what is the reasoning behind why you just have chosen so short periods after application of the salt?
Yeah, so the reason was that I want to see the direct expression patterns after the salt treatment. So how the plant adapt to the salt treatment, which transporters are upregulated or downregulated. So this was the idea to directly use this time in 3 days after the treatment started. I don't know if this was a question, but
no, that's maybe you can add that we already did similar experiment with the long-term, let's say, long-term harvesting times after salt application for Salicornia, for example, so that we have already some experience. So it's a different approach, might be also interesting, but you focused now on the short-term effects. Yes, thank you. So I think there are only now 2 thank you messages. Are there any more questions? Well, maybe a question from my side. I mean, you were talking about these differences in the response to sodium chloride and sodium sulfate. And I think you're also planning to test other salts. Yeah, I think you mentioned in your last slide. So what is your expectation, or in what kind of environmental conditions you would even contamination or increase of other salts in the soil? Was my question clear?
I hope so. So the idea is, so you want to know what's the effect, this toxic effect, or what's the reason for this? Or
toxic effect of different salts, actually, and also in which part of the world you might actually have this influence of other salts. I mean, it's mainly sodium chloride, but in Argentina, you're told that it's also sodium sulfate. Yes.
So one example is Argentina. There are both salts in the soil. So it's always important that you compare different salts. As you can see, there's a toxic effect to sodium sulfate, and you need a plant which can survive all these different kinds of salts. And for example, fertilizers during irrigation systems can also accumulate other salts like sodium chloride. And this is the reason why I want to test different salts and not only sodium chloride. And the idea why I used sodium sulfate and potassium sulfate is that at the moment we don't know if this toxic effect is due to the sulfate or it's due to the sodium. And with this treatment, the idea is to find out if it's the sulfate or is it the sodium.
Yes, thank you. Then there's one question in the chat who is interested why you work with hydroponics and that it might be better directly to switch to substrate or soil. Yes, because maybe it does not completely reflect the natural situation, and also, of course, you have a different rhizosphere, different microorganisms, of course. So what is your answer?
This is definitely a point. The reason why we used hydroponics is that you easily can apply this salt solution into hydroponics, and it's a very controlled condition. So you know what is the composition of all other elements inside the solution, and in soil you never know how strong these salts are bind to the soil particles, if it's available for the plant. And yeah, but it's a point of maybe in the future you could also try to make some experiments in soils, but for the beginning we decided to use hydroponics because of these controlled conditions.
Yes, thank you very much. So, are there any more questions? Then please ask now, because otherwise our time is it's a response. Working with hydroponic system could not reflect could not reflect the profile of salt accumulation in the rhizosphere and could affect gene expression. It would be better to use substrate. It's the same question, yeah. Okay, so I cannot see more questions right now. So I think it was a very successful start of the webinar series. I hope you agree. And maybe I can advertise that all working groups in the Sustain Action will have their webinars. I think the next one will be in July, but please follow our homepage or also via LinkedIn. You can find some information about the next seminars, and would be great if you maybe also join webinars of the other working groups. You are highly welcome. Yeah, and thank you very much again for joining, for organizing as well. And I mean, Kate, do you want to say final goodbye?
Thank you very much, Jutta. Also, big thanks to both speakers. I think you did fantastic, and it was also very interesting to hear your speeches. Also, big thanks to Syed for organizing, and to you, Jutta, as well, for leading the webinar. I have just copied the link to the webinar series that we will be having this summer and early autumn. Thanks a lot for joining us today, and we hope to see you next time as well.
Thank you. Bye. Have a nice
Bye-bye.
day.